Hi guys, my name is Tim. Can you all hear me okay? I'm a sophomore. I just started my research this summer and I work in Dr. Arribas' lab studying developmental biology. Okay, so to start, did you guys know that not all of your genes are on at once? Okay, if you didn't, then this is going to be interesting. So your genes are constantly being turned on and off during development. Specifically in regions of DNA called enhancers. Enhancers are not genes, they're everything kind of outside of the genes. So enhancers have specific proteins called transcription factors that bind to them in a certain sequence. And when that sequence is set up in a specific way, it drives the gene. So it kind of works like a password, requires specific characters, letters, numbers, etc. I'm generalizing this. So the best way to study this is in fruit flies. Why? 60% of our DNA is like fruit flies. Nobody cares when you mutate a fruit fly, and we can murder hundreds of them and nobody cares. So when the fly starts off as a single cell, develops into an egg, a larva, some people call it a maggot, a pupa, and then the fly and this whole thing starts over again and your trash can is buzzing with them. Okay, so we picked to study specific enhancers in the larva wing. So larvae don't actually have wings, but they are developing them, so they're inside here and they're turning into wings over time. We did a computer search to find enhancers that are working in the developing fly wing. We came up with about 100. I don't have time to get into all of them, but here's a pretty picture of 4 of them. And I'm just going to talk about 2 today. Before I get started though, I should probably explain them a little more closely. So the blue is a protein called DAPI. It just lights it up and makes it look nice and pretty. The green is the actual enhancer, telling the gene where to go. So anything that's lighting up green is saying, hey, gene, you are allowed to be here. And then this purple pink stripe here is essentially cutting the wing in half so we know where everything's being expressed. Okay, so the first enhancer that we discovered was called the NAB enhancer. And this is its sequence of DNA right here, and here are the transcription factors that are making the password. Very similar to this password for UNC. However, they look completely different looking at the green. So how do we know why is it doing this? We have to mutate specific protein binding sites to find out. So we destroyed a protein binding site called SUH, and in one instance for NAB, it completely turned off the expression. For UNC though, it just lit the whole thing up. Why? Well, enhancers are a little more complicated than just, oh, have all the letters in the right combination and everything works fantastic. Certain things need to be next to each other. There's spacing, there's number of them. When you put an S makes a S sound, H makes a H sound. Together they make sh. Kind of makes sense, get what I'm going at here? So we study enhancers to understand how mutations are working, how proteins are functioning, and one day we'd like to actually just make our own.